Welcome back to part 2 of the DNA biotechnology presentation and lecture. And the last time I left you, I was explaining to you in part 1 just a little bit about how you can actually take parts of a chromosome, sections called genes, and you can actually insert them into a bacteria's circular piece of DNA called a plasmid. And today what I'm going to quickly talk about, let me get this set up here, is I'm going to talk to you guys a little bit more about what you can do, like why this is important. And I talked a little bit about that in the other video when I was talking about medicine, but I'm going to show you that a little bit more right now. Tons of uses. What the uses is to clean up oil spills and any kind of toxic waste spills that happen in the environment or into waters. It can help make pesticides, which are chemicals that help fight and kill insects so that they don't destroy crops that we need to grow food. You can also use it to make, like this is what I was saying in part 1, is to make certain kinds of medicines and drugs that can help humans better able to be adapted for something like for cholesterol, for example. And I think I might not have talked about this one in the video, but we can also use them to make vaccines, to make genetic material that can fight off diseases for us. And last but not least, one of the things you can do is you can make what we call GMOs, or genetically modified organisms. And that essentially means that you can take an organism, let's say a tomato, and you can change its DNA and insert genes into it that will allow it to become a bigger and better tomato. And it grows better, and just because you insert 1 or 2 different genes that help it grow better. So that's called genetically modifying or changing organisms. And so you can get bigger and better fruits, or you can get a chemical that works better, or you can get an insecticide that is really resistant to any bugs eating the plants and stuff like that. So you really can help make organisms that are more adapted, and it helps humans. And like we said, that's called biotechnology, the use of living things to help humans. And as I mentioned, if you somehow are an organism that gets a gene put in you that's not initially your gene, so it's an artificial, not your own gene, that's called a GMO, genetically modified organisms. And I'm going to show you a picture in a second. And any organism that is a genetically modified organism that has DNA put into it from a different species, like a bacteria having a human gene put in, we call that transgenic. Transgenic means the genes are going across organisms. I'll give you a second to take a couple notes. And I'll talk to you in a second. This is also very easy to do with plants. So if you want a plant that has bigger leaves, or a plant that has bigger corn, or a plant that has better tomatoes, you can do this to plants too. And this is the picture that really kind of explains all of it. Let's just say here you've got a piece of DNA from some organism. Let's say it doesn't really matter what kind of organism. You take the gene out, like we talked about in the last part. Then you grab the plasmid from the bacteria that we talked about in part 1, and you insert it with those enzymes, and you put it into the plasmid, and you put it back into the bacteria, and it has that gene, that particular gene in it. And the bacteria then multiplies and makes a lot of that chemical. If you look real closely here, you can actually see that gene in all of these bacteria cells. So if that particular gene was a chemical that kills bugs, you could spray it on the crops, and the crops will be protected from the bugs. Or if that chemical helps the bacteria clean up and digest toxic or dangerous materials like oil spills here, you can actually make the bacteria, and then you literally spill the bacteria into the water, and they'll eat the oil up so that the fish aren't damaged. Here's another example. I actually take a medication for this, where you take a gene that makes a protein, and you make copies of it, and then you actually can give it to somebody, like a human, and it's one that dissolves blood clots in the body, and other things that might build up in the middle of your in your blood vessels, so that your blood can't get through your body. So there's medicines that you can make for that, and all you need is a bacteria with a plasmid, and you need a piece of that DNA that has the gene on it that you want. One of the other applications that I think that you guys might find interesting, if you're ever, I'm not really much of a skier, but if you go skiing a lot and you go to a place that has fake snow, not real snow, that is actually made from a protein that's taken out of an organism, put into a plasmid from a bacteria, multiplied many times, and then that protein then gets sprayed all over the hills, and it's actually a fake snow. and it forms at at a higher temperature. So it's kind of real snow. It's water, but the protein that you use helps turn it into snow at 55, 60 degrees, so you can still go snowing even when it's not actually snowing outside. So again, this mentions here all the different reasons why we would want to take a gene and put it inside a bacteria. This multiplying right here can take literally a matter of hours. It doesn't take very long at all. We do this with insulin, for example, to help people with diabetes. We put the insulin gene here, we make lots of copies of it, and then we bottle it up and we sell it, and people who need it can buy insulin. And I mentioned the plants are good for this too. So here's a bacteria plasmid, and here's the bacteria cell. And you're taking a gene out of the plant that, let's say, makes the plant have bigger something, leaves, or better, stronger leaves. And you put it into the plasmid, and you multiply it. And then there's that gene, and you get a plant cell that has that gene in it, so the plant grows. And then the last thing that you've probably heard about that has a lot to do with genetic engineering is cloning. Cloning is basically something that's been done with plants for a long time. With animals, it's obviously brand new. It's a very hot topic, a very debated topic. It's very controversial because in this one, instead of taking one gene and putting it into a bacteria cell, you're actually going to take a nucleus of an animal cell and you put it into an egg of a female of that animal, and it actually makes a clone of the female. A clone, if you don't know what that word means, is an identical copy. Not just an identical, it's kind of like an identical twin, except that it's an identical twin of the mother. So it's the mother's copy. They've done this with sheep. And of course, there's lots of problems with that because, if somebody was able to do that all the time with no restrictions and no medicine and no doctors, there could be a lot of people trying to make copies of themselves, which would be kind of weird. And I'm not going to talk too much about gene therapy, but gene therapy basically is when you take something like part of a DNA or an RNA and you take out the gene and you put a healthy gene in so that the body then you can inject it with the syringe there, and then when you put those cells in the body and they multiply by mitosis, you get a lot of cells that have that gene that's a healthy gene, and it makes many, many copies, and you can actually repair too, and that's called gene therapy. So an absent or not working gene is replaced by a normal gene. And then one is called germline therapy. You don't have to know the name, but it's when you change the sperm and egg, the gametes. And then if you actually are changing and injecting into the actual body cells, that's called somatic gene therapy. Well, my kids are home, and I'm going to say goodbye for now. So I hope you guys will move on to part 3, which is about a certain thing lab that we're going to do. Have a great night, guys. I'm being called by my 4-year-old. Bye-bye.